Dr. S. Uma Maheshwari, Associate Professor and Head, Department of Zoology, Thandai Periyar Government Arts and Science College. Today I am going to deal about cell cycle. We all know the growing cell undergoes cell cycle, and the cell cycle is divided into two distinctive phases, interphase and the phase of division. During the interphase, the cell spends most of its lifetime in the interphase, and this is the period of intense biological activity, and there is duplication of the chromosome complement. Some of the differentiated cells like the mammalian cell, nerve cells, divide rarely or do not divide after birth. Human neuron, that is, it stays in the interphase and lasts the entire lifetime of a person. Thus, cell cycle is a complex phenomenon by which the cellular material is divided equally into the daughter cells. The cell cycle has been divided into four successive intervals as G1, S, G2 and the mitotic phase. During the G1 phase, the DNA synthesis starts. The G1 phase is the period between the end of mitosis and the start of DNA synthesis, and S phase is a period of DNA synthesis, and G2 phase is the interval between the end of DNA synthesis and initiation of mitosis. A mammalian cell growing in culture with a generation of 16 hours is represented by a G1 phase of 5 hours and S phase of 7 hours, G2 phase of 3 hours and mitosis phase of 1 hour. Depending upon the physiological condition of the cell cycle may last for days, months or years. The mammalian nerve cells and the skeletal muscle cells and the rarely dividing the circulating lymphocytes contain the amount of DNA that is present in the G1 phase. If you compare the normal cells and the transformed cells, the normal cells when cultured in the media exhibits inhibition, growth inhibition, that is contact inhibition, and also stop at the G1 phase, whereas the transformed cells divides continuously and pile up into layers. And there is also a G1 state in the G1 phase. During the G0 state, the cell may be considered to be withdrawn from the cell cycle, and when the conditions change, the growth is resumed and the cell re-enters the G1 phase. That is, the G0 phase is a checkpoint. The eukaryotic chromosomes undergo condensation and decondensation cycles at the cell division. The interphase. During this phase, the chromosomes are decondensed and synthesis of RNA takes place. Highly condensed DNA cannot be transcribed. Perhaps DNA cannot be reached by RNA polymerase. Molecular events that occur at defined stages of the cell cycle includes the DNA synthesis, as the S phase sets where DNA synthesis lasts for several hours and many replications are sequentially activated. In all these cells, the more condensed heterochromatic regions of the chromosome replicate late during the S phase. Centromeric heterochromatin replicates later than the rest of the chromosome. If you take the mammalian female, the X chromosome that becomes inactivated is heterochromatic and late replicating, whereas the one that remains activated in euchromatic is euchromatic and replicates earlier. So if you see, there is a decondensation-condensation cycle here. During the G1 phase, the chromosomes are completely dispersed, and during the S phase, the duplication occurs, and during the G2 phase, the condensation starts. And during the metaphase and the anaphase, we can observe the maximum condensation of the chromosome. The molecular events that occur during the cell cycle, the S phase, here the histones are synthesized, and during the mitosis phase there is decrease in the protein synthesis and lower level of cyclic AMP is observed. The arrest of DNA synthesis and RNA turnover take place. The eukaryotic replication occurs in the GC-rich region of the DNA, and appearance of inducer factor for DNA synthesis is observed. Histone synthesis begins. In the heterochromatic replication, 80 richer DNA sequential initiation of banks of replicating units is found. In today's class, we have seen the sequence of events that leads to the distribution of the cellular material equally divided between the daughter cells. Thank you.